Okay, so an antibody. What is an antibody? It is a glycoprotein molecule which is produced by the plasma cells in response to the immunogen to protect host against the extracellular pathogens. It is made up of 4 polypeptide chains which are too heavy and too light. There can be some exceptions. It is a part of the adaptive immunity. It binds to the region on the antigen which is called the epitope, which is the antigen determining region. It is 20% of the proteins in plasma. It can be monoclonal and it can be polyclonal as well. So it has two ends, the Fab end and the FC end. The Fab end is called the antigen binding fragment and the FC end is the crystallizable or the constant fragment. The Fab end includes variable region of the heavy chain and the light chain, and the constant region of the light chain as well the constant region of the heavy chain. It is 5 to 10 amino acids long. The antigen and the antibody binding includes the forces which are van der Waals forces, electrostatic, hydrogen and hydrophobic bonds. Specificity of antigen is due to the hypervariable region between the heavy and light chains. The constant fragment includes the remaining of the 3 or 4 constant regions. Its functions are to attach the antibody to the host cell. Region, it is a region to fuse the IgM and the IgA into multimers. It is necessary for the transport of Ig across the epithelial barriers and the IgG across the placenta for it to pass from mother to the fetus. It has 5 classes which can be learned from the mnemonic game D. The IgG is always present and it is the most abundant. The IgA, it is present in the secretions and blood and intestine. And the IgM is the first antibody produced in response to any immune reaction. The IgE releases histamine and the IgD is present on the B cell surfaces. So there are 2 types, the heavy chain and light chain. The light chain has 2 types, kappa and lambda, which are present in the ratio 2:1, and if this ratio is disrupted, it is disrupted in malignancy as in multiple myelomas. The heavy chain includes alpha, delta, gamma, epsilon and mu. It can be remembered from G, gamma, alpha, mu, epsilon and delta. The functions include neutralization of the virus or any toxin, agglutination, activate complement system, lysis of the virus or any microbe, opsonization, ADCC, which is an antibody dependent cell mediated cytotoxicity. So this is the structure. There are 2 light chains and 2 heavy chains. It is a Y-shaped structure. It is a glycoprotein. There are 4 polypeptide chains. The light chains have 1 variable and 1 constant regions. The heavy chains have 1 variable and 3 constant regions. The Fab fragment includes the variable and constant regions, and the FC receptor and the FC region includes the constant regions of the heavy chain. And there are disulfide bonds between the 2 heavy chains and between the light and the heavy chain. The upper end is the antigen binding region and the lower end is the FC receptor binding, and it is a carboxy terminal end. Thank you.